So we are going to, can you all see my screen?
Yes.
So we are going to continue with microbial genetics. And last week we already saw, exchange of genetic material between bacteria. And we talked about F factor, fertility factor, how F prime plasmids are generated, how F prime plasmids are used in bacterial genetics to understand bacterial genes. I must mention that these slides or the content on these slides in microbial genetics, what I'm using is coming from lectures. So there are few lectures on MIT about in the open courseware, and I have selected topics which are best for sophomore genetics at your level. So you can consult for additional questions. You can also consult those ones as well. So we are going to today cover two things. One is how can we do the genetic analysis in bacteria using transposable elements, and also how can we play with the phages, bacteriophages. So in last two lectures, in the last lectures, we saw we talked about conjugation, in which we talked about fertility factor, and we also saw F prime plasmid. Then we also learned about how F prime plasmids are isolated, how they can help us in the genetic analysis in bacteria, for example, complementation, or to determine mutant phenotype as dominant or recessive. Today we are going to look at transduction, and also we are going to see how can we use transposable elements in genetic analysis in bacteria. So if you recall, the different transposable elements in bacteria were like TN3, TN5, TN10. So here you see a TN5 transposon, which contains kanamycin resistance gene, as well as a transposase gene. And then you have these inverted repeat sequences. Okay. So any bacteria which harbors TN5 transposon, it will be kanamycin resistant. That bacteria, when you will grow on, for example, LB plus kanamycin antibiotic, you will see colonies if you have used TN5 to grow that bacteria. How can we use TN5 for genetic analysis? How can we use TN5 to do mutagenesis, and how subsequent analysis after mutagenesis with TN5 is performed? This is what we are going to see. Think of process of mutagenesis using P element in fruit flies, or for example, in plants, agrobacterium mediated transformation, the tDNA mediated insertional mutagenesis. So in similar context here, we are going to generate mutant population. So before we go into the mutant screen in fly, it's important to understand that bacteria with special genotype are used, as well as the phage, because this transposon is to be delivered inside bacteria. And this delivery, for this delivery of transposon, we are going to use phages. Okay, so there has to be a special genotype of the phage, and also genotype of bacteria. What you know already that bacteriophages are viruses which infect bacteria, and this phage, it injects its DNA into bacteria, and once inside the bacteria, phage DNA can get integrated into the bacterial genome, or it can also stay as a plasmid, as extra chromosomal plasmid. So in order to deliver TN5 genome into bacteria so that we can do some mutagenesis of bacterial genome, we use special phage vector, and the genotype is lambda phage, PAM, int minus, and TN5. We need to understand what PAM is, what int minus is, because we already know TN5 is a transposon, which is going to give kanamycin resistance. So PAM, int minus are basically, so int minus is phage. It lacks the ability to integrate into bacterial genome. So we have bacteria, phage, this phage with int minus mutation. When its DNA will come into the bacteria, it cannot integrate into bacterial genome. It will remain as such. However, the other one is also very important. This PAM mutation, it allows conditional phage growth. These phages, they were actually these mutations in bacteriophages. They were very specialized one. For example, when lambda PAM phage infect E. coli with AMBER suppressor. Now, what is AMBER? You know the stop codons? Stop codons are UAG, UGA, and UAA. Now, UAG is AMBER, and then I believe this one is ochre. I explain you in a moment why they are amber, ochre, etc. And UGA was named opal, or yeah, opal, I believe. Now these phages, they were discovered to be infecting only certain strains of bacteria, because phage with this PAM mutation, it can only infect and multiply when bacteria contains AMBER suppressor mutation. Now, normally what we have an open reading frame, AUG, and then you have UAG, the stop codon, the open reading frame. Let's see, you have acquired a mutation, which is a nonsense mutation, which means a stop codon is appearing much earlier, which means this open reading frame is disrupted and protein will not be formed. Translation will stop right away by making a truncated protein. And if it is an essential protein, organism will not grow. So normally each of the codon, they are read by tRNAs, and tRNAs, they bring in a corresponding amino acid. For example, AUG will bring in methionine. And similarly, at the stop codon UAG, there are no tRNAs which can add corresponding amino acids, because these are the stop codons, a signal for ribosomal translation machinery to stop translating the protein and mature protein is or polypeptide is, uh, complete is completed here. Now, phages which contain MBI mutations, for example, in a very essential gene here, these phages, they cannot grow in normal bacteria. Normal bacteria means which will carry normal tRNAs, which will recognize it as a stop codon, and phage protein will not be formed. And since this phage protein is essential, phage will not be able to multiply in suppressor minus mutation in bacteria which lack such a mutation. However, these phages were discovered in mutant screens, in which they discovered that this phage is normally not able to grow in E. coli, but in specialized mutant bacteria which contain suppressor mutation, UMBI suppressor mutation, they were able to grow. What happened? These bacteria, they do not recognize this as a stop codon, this one, and they add an amino acid here. A tRNA comes in, and they, instead of recognizing it as a stop codon, they add a a specific amino acid. That's why it's called UMBI suppressor. UMBI suppressor. So bacteria has a mutation which does not recognize this as a stop codon, and then this protein is synthesized and phage is able to grow in these bacteria. So what we have now, we have lambda PAM phage, which carries some MBI mutation. Also, it carries integrase mutation, so that the transposon, it cannot integrate into the bacterial genome. And also we have kanamycin resistance transposes. So I explained you what int minus is and TN5 is. So let's say we are interested in screening bacteria for, in the yeast, we also did, histidine synthesis. So let's say we are doing histidine synthesis in bacteria. You are interested in genes which are involved in histidine biosynthetic pathway. We use bacteriophage, which carries this genotype together with TN5 transposes. And when this will infect E. coli, which carries, which is sensitive to kanamycin resistance, and also which does not carry MBI suppressor mutation. Okay, two things. We are infecting E. coli host, which is sensitive to kanamycin, as well as it does not carry phage MBI suppressor. Now, what will happen? Phage will not be able to replicate because there is no MBI suppressor, nor the phage DNA can integrate into the host genome. So phage will infect, DNA of phage will go inside the bacteria. Due to lack of MBI suppressor, this DNA does not replicate, as well as it cannot integrate into the genome. Since this bacteria is kanamycin sensitive, if we now grow these bacteria on kanamycin plates and screen for kanamycin, it means this bacteria can only become kanamycin if TN5 from the phage DNA has jumped from phage genome to host bacterial genome. Clear?
सही जी, आपने लास्ट पॉइंट रिपीट, लास्ट पॉइंट दोबारा से रिपीट कर दें।
जी मैं अभी करता हूं. So let me draw here to make things even more clear. So we have phage, it injected its DNA inside the bacteria. This bacteria is kanamycin sensitive. Also, it is suppressor minus. It does not encode M-by suppressor. Phage DNA, when it comes in, we know it contains TN5, which also contain kanamycin resistance gene. Since due to lack of M-by suppressor mutation, as well as int being int minus, which means phage DNA lacks the ability to integrate into the genome, the only possibility to make this bacteria kanamycin resistant is that this kanamycin gene, because it is a transposon, it jumps from phage genome to host genome, and bacterial genome now carries TN5. Clear, Varis?
जी सर, तो सर ये सिर्फ एक चांस है कि TN5 वहाँ पे जाके मूव कर जाएगा, जरूरी नहीं है कि वो मूव करे।
Yeah, it's a chance. So there will be 1 in 100,000, I believe. Yeah, 10 to power 5. It's 100,000. Am I right?
जी सर।
1 in, it's a chance. 1 in 100,000. 1 in 100,000 means we will have 100,000 phage bacteria, 100,000 bacteria infected with phage particles. Out of those 100,000, one such event we will recover. This is the probability. Clear?
Yes sir.
Okay. If we say bacteria E. coli contains 4,200 genes, we are interested in finding histidine pathway or biosynthetic pathway genes, all the genes involved in histidine biosynthetic pathway. And our probability is 1 in 10 to power 5. We need to calculate how many bacteria we should infect to make sure we possibly get every possible gene out of these 4,200 infected. So if we do this calculation, we'll say we will infect 2 into 10 to power 9 E. coli cells with lambda phage, the one with TN5. And if we calculate 10 to power 5 bacteria will get 1 TN5. Out of this, 2 into 10 to power 9, how much we will get?
20,000.
Like this. Am I right? 2 into 10 to power 4, 20,000. Is it clear to all?
Yes, sir.
Yeah.
Good. Now, how much is the total number of genes in bacteria? 4,200. How many more we have? 5 times, nearly 5 times more mutants. And we use more than 5 times or like near 5 times because we want to make sure, because these events are random. These are not, TN5 is not going to jump in only your histidine. It is going to randomly jump all over in the bacterial genome. So we use 5 times more so that we become confident that we have more or less every spot in the genome of bacteria, we have a transposon. What we do after this process, we select bacteria on kanamycin. And what we have now, 2 into 10 to power 4, 20,000 kanamycin resistant colonies. And our idea is that more or less every gene in bacteria will be at least mutagenized once by TN5. Now, we should also know that in these 4,200 genes in bacteria, there are many genes which encode RNA Pol2, which is RNA, not RNA Pol2. Pol2, when we say 2, it's a eukaryotic. So in bacteria, RNA polymerase machinery, which is involved in transcription. There are also ribosomal genes involved in translation. Since these are critical, extremely important, we will not find mutants for them. They will die. Okay. So it means the ones which we are recovering, 20,000, if we exclude them, our chances of saturating the genome is even high. Okay. Now, how are we going to screen for histidine mutagenesis, histidine mutants? I would like you to recall what we did for yeast. In the yeast, you remember we did replica plating. How many of you recall replica plating? Anyone remembers what replica plating was? No? So you recall what we did, we said, and we will do same thing here. We will grow E. coli, what we have 2 into 10 to the power 4 strain, but mutants after, we will spot them individually on plates on a grid like this, very precise location. And we use, let's say, 100 bacteria, independent bacteria on one plate. How many we have? 20,000?
Yes, sir.
No, not 20. Yeah, multiplied by 2, 20,000. So if on one plate we have 100 bacteria, how many plates we will have for all 20,000?
200.
200.
200. And if we double the number of bacteria, if we say, because we have to minimize our job, our work. If we double the number of bacteria, let's say we make them 200 bacteria, how many plates we will have?
100, sir.
100. So we have reduced the work by half. We'll have 100 plates on normal media, which contains everything, and these bacteria can grow. Now we will replica plate. We'll take a stamp. Now replica plating should be clear to you all. So we take a block, and let's say this is my palm is the plate. Size of our block should be exactly the size of the plate. Normally small plate is 90 millimeters or so. We cover this plastic block with a velveteen cloth, and then come on the plate like a stem, and we lift, and by lifting we'll see on the velveteen cloth all 200 colonies which are spotted on a grid here, they are imprinted. and we go with this stamp onto replica plate, which is this one, which lacks histidine in the media. Here, everybody will grow, but when we will replica plate, we will see gaps. Where we see gaps, these ones are histidine mutants. They cannot synthesize histidine and they cannot grow because in the media we lack histidine now. This is like a minimal media. Do you recall it now?
Yes, sir.
Yes.
We did this in yeast auxotrophic screens. Now, what we have recovered, we have now histidine mutants, which are result of this mutagenesis in which phage injected its DNA. DNA contained TN5 transposon, and transposon has jumped into bacterial genome and it has inactivated or mutagenized histidine biosynthetic pathway genes. You can screen the same number of colonies because these are your master plates. These plates are with you and you can screen them. These are mutants. You can screen them for histidine biosynthesis. You can screen them for bacterial motility. You can screen them for lactose utilization. You can galactose utilization, carbon source utilization, antibiotic resistance, whatever. Once you have a mutant population, this mutant population can be screened for as many questions which come to your mind. Okay. Once you identify these, let's say you identified out of these 20,000, you have identified 50 mutants, independent mutants, which show defect in histidine synthesis. They do not grow on minimal media. Now, you need to isolate the gene. If within the bacteria we said, there are 4,200 genes, and you have 50 independent mutants, if this is the bacterial genome, simply by doing PCR, and you remember we talked about this one PCR in flies as well, we called it inverse PCR. We know the sequence of TN5. We know sequence of inverted repeats here. This one and this one. Once we know the sequence, and we are assuming that this TN5 has jumped into the host genome, if we know this sequence, we can simply design a primer and do a PCR, and our PCR will give us the flanking sequence in the E. coli or bacterial genome. Let's say TN5 is sitting here. We have a primer which goes, because it's an inverted repeats, single primer we will use. It will go on this side as well as on this side. We'll use random hexamers which will randomly bind on the genome. And this PCR product which we'll get it sequenced. Once you know the sequence of this PCR product, you will compare with the bacterial genome, and comparing the bacterial genome, you will get the flanking sequence of TN5. And this flanking sequence will tell you which gene in bacteria is precisely disrupted or mutagenized by TN5 and has caused his minus phenotype. Is it clear to all? Is it clear?
Yes, sir.
बोल दिया करो यार, yes, no. So there are limitations for transposon mutagenesis because usually, they like to get uh, conditional mutations as well as nonsense mutations. Nonsense mutations, these are premature stop codons, which are very, very strong mutations, and also temperature sensitive, so that you can, even if a mutation is lethal in normal temperature, let's say at 37, even if there is a mutation in bacteria, it will grow just like a normal bacteria unless you shift the temperature to restricted temperature, and only then bacteria will show some phenotype. So you cannot acquire these kind of mutations using transposons, because transposon insertion, it completely inactivates a gene and it may be lethal. And if your mutation is lethal, you cannot recover mutant. That will not be even visible on kanamycin plates, because that mutant bacteria is simply dead. So the other strategy they have developed, the bacterial geneticist, is the transduction. Transduction is a very powerful tool, very intelligent tool. It has its own limitations as well, but as compared to TN5 mutagenesis, where, based on insertion of TN5, you acquire kanamycin mutagenesis, and then you screen for the gene of interest, which many people have actually used. TN5 mutants in the population generated by such jumping of TN5 has been used extensively, but they refined the bacterial genetic analysis using transduction as well, in which bacteriophages not only just used to deliver its DNA, but just like we saw, that will be a wrong analogy, but still you can think of, the F prime plasmid getting integrated and then outlooping, excising, although it's actually bacterial own genome. A similar kind of strategy is used with phages, because absence of meiosis in bacteria is a problem for conventional genetic analysis, which we normally do in eukaryotes. Also, to circumvent, to overcome lack of meiosis in bacteria, there are still tools developed which can be used to do complementation test. Last time we talked about F prime plasmids, also mapping of genes, how distant, how much away genes are from each other. You will see today one of the tool using transduction again. So how do we perform genetic analysis using transduction, where bacteriophage genome is involved? For example, E. coli genome, this is a total size of the E. coli genome. 4 million, a bit more than 4 million base pairs. Okay, 10 to the power 6 is 1 million, and 4.6 means 4.6 million base pairs. The bacteriophage P1 chromosome is size of bacteriophage's genome is 10 to the power 5, which is 100,000 base pairs. If we compare, so this is size of bacterial genome, this is size of phage genome. And we talked many times about, lytic cycle and lysogenic cycles of the phage. Normally, phage goes into the bacteria. Phage DNA is injected by the phage, where, many phage molecules are generated. Once its genome is transcribed and translated, and without integrating into the bacterial genome, it can make many of its, because its genome gets transcribed, translated, its proteins are assembled, and then it packages its genome. It bursts open, kills the bacteria, and many phages are released. These phages then go and infect other bacteria in surround neighboring environment. The other cycle, and this cycle is called lytic, where lysis is involved. The other cycle is called lysogenic life cycle, in which the bacterial genome is invaded by phage genome, and phage gets incorporated or integrated into the bacterial genome, and from here it can transcribe, translate it, and again phage particles can be made. But in this case, it does not kill bacteria. It uses bacterial genome for its own survival. It doesn't kill, because in this case it knows I'm residing myself within the bacterial genome. If I kill it, my own genome is going to be lost as well. But whenever phage wants, it can hop, it can outloop or excise itself and go back to lytic cycle as well. Now, this is used, this intelligent tool, which is, bacteriophage arsenal, is used to do transduction or bacterial or mutagenesis in bacteria for genetic analysis. This is what I just explained you. Normally, this is the phage DNA. We learned in last lecture as well. It replicates through rolling cycle mechanism. Okay, and when complete genome is replicated, as soon as it gets complete, it gets cut, and complete 10 to power 5 base pairs of phage genome is packaged in this head. And then the next 10 to power 5 base pairs, they are packaged. Then, since this is a rolling cycle, is like ribbon is being pulled out of the pulley, and it's being replicated. The replication machinery is making copies, and in the form of concatenamers, it gets cut precisely 10 to 5 base pairs and packaged into phage head. Now, there are errors sometime, and 1 in 300 phages, by mistake, it can package E. coli chromosome. Normally, it does not happen. 1 in 300 phages, they can by mistake package, instead of their own genome, they can package chromosomal DNA of E. coli. And if we calculate 1 in 300 and how much is the bacterial genome, 1 in 300 by mistake can carry phage genome, and how much is the capacity of phage head to package is 10 to power 5 total. So it means each phage, if we calculate the probability, 1 in 300 bacteriophages may carry E. coli, and then the size of the genome. So we'll say it's nearly 1 in 15,000 phage will carry a particular E. coli chromosome. Because if 1 in 300 contains bacterial genome, now that bacterial genome could be this region, it could be this region, it could be this region, it could be this region, this. In order to see whole of the bacterial genome packaged in phage particles, we have to have a huge population of phage particles. And the probability that phage will contain bacterial genome will be a complete bacterial genome will be 1 in 15,000 phage particles. How all this is incorporated in genetic analysis, how they use bacteriophage as a genetic tool for genetic screen and uh, to ease the genetic analysis. What you can do, let's say you grow phage, P1 phage, on wild type bacteria whose genome is A plus B plus. You have mutants for A minus, B minus already, and these mutants could be TN5 transposon driven mutants. You know the phenotypes, but you don't know the physical distance between A minus, B minus. You don't know whether A minus, B minus are dominant or recessive because bacterial genome is haploid. What do? You grow P1 phages on wild type bacteria, and your probability is 1 in 15,000 will carry A plus or B plus or whatever, or other genes as well. Because when phage by mistake carries bacterial genome, it not necessarily is going to carry A plus or B plus. It can carry any of the other genome parts. Now you will infect, so these phages, these ones, these P1 phages, which infected wild type, and then you isolated all these phages, you infect now A minus B minus bacteria, which you already had, with these phages, which may carry A plus B plus. And what do, you now screen for A plus phenotype or B plus phenotype or A plus B plus both. A plus could be, for example, histidine. It was his minus. This one, for example, is leucine minus. These are the phenotypic screens or genetic screens you already did. Now, for example, so this is your bacteria, which is A minus, B minus, okay? You infected individual phage particles, okay? You designed in such a way that, and you selected this one after infection with P1 phages, which may have A plus B plus. You screen for A plus phenotype. You know this bacteria is His minus. You select it on media which lack histidine, and you try to see if it has become, this His minus has become His plus, which means wild type. You select same bacteria for B plus phenotype, which is, let's say, leucine minus, and you try to see if this phage has brought in leucine gene, wild type, because this was wild type. If you find A minus, which is His minus, is rescued by phage and it has turned bacteria into His plus always, but it is not making it, so it's never becoming A plus B plus. Individual, a different phage is giving you A plus and a different one is giving B plus. You will conclude that A plus and B plus are far away because how much is the packaging limit in bacterial genome is 10 to the power 5. It means A plus and B plus are really far away from each other. And that's why you don't see A plus B plus together. However, if they are together, then closer to each other, and in one phage head, which was 1 in 15,000, if they were close enough, this A plus B plus is brought in by the phage. Since A plus B plus is closer together, there is recombination. Now your bacteria, which was His minus, leucine minus, or A minus, B minus, is converted into A plus, B plus. And due to this phenotype, now you will say A and B are close to each other. We express it in such a way, it is expressed in such a way, if you have 100% co-transduction. You remember transduction is when phage particle is involved in transfer of DNA from one bacteria to another. So when co-transduction is 100%, which means A and B, A plus B plus, are always going together, and we are always seeing His plus, leucine plus, or A plus B plus phenotypes in our A minus B minus mutants, we'll say they are close to each other. The distance between A and B is less than one phage, one headful. One headful is 10 to the power 5 base pairs, which was size of P1 phage genome. I remind you this one. 10 to the power 5 is size of phage. So if A and B always co-transduce, it means they are close enough in the genome. That's why phage is always packaging together in the same phage head. But if they are at a greater distance than 10 to the power 5, we will not see co-transduction. This is equivalent to bacterial, equivalent to two-factor cross of eukaryotes. Is it clear so far?
Yes, sir.
Okay. Now let's see bacterial three-factor cross. We have now a third. The 3-point test cross was, you remember, in plants and eukaryotes, we did 3-point test cross was where we have 3 independent phenotypes, and we set up a test cross with a triple mutant and then try to map the genetic distance. Similarly, you have now included a third mutant phenotype. Let's say you have His minus, leucine minus, and tryptophan minus. Okay. We infect P1 phage to A, B. So His minus is A, leucine is B, and tryptophan is C. After infecting bacteria, you isolate the phage particles. These phage particles, you infect A minus, B minus, C minus bacteria, and you then, after infecting, you screen these bacteria which are infected with them for, let's say, A plus phenotype, A plus B plus, A plus C plus, B plus C plus, A plus B plus C plus phenotypes. Now, if A, B, and C are less than one headful distance, which is 10 to the power 5, we will find A plus B plus C plus co-transduced always. But if distances are different between A and B, B and C, we will not see co-transduction always. because it depends on the physical distance between A, B, and C. If they are farther apart, more than one head full, we will find different phenotypes. Maybe we get A plus C plus. Maybe we never get A plus B plus. So based on the phenotypes, let's say we get A plus B plus C plus, which is this one. This means there's double recombination here and here. Thus, distance between A, B, C is less than one phage head full. It can be complicated also because, even if it is less than one phage head full, it also depends on the recombination, where recombination has taken place. Even if they are one phage head full, you may get A plus B plus, which means the two crossovers are here, A and B, and they're excluding C. But if the two crossovers are here, we will get A plus B plus C plus. Or it may just be here, A plus and not B and C. So this is how the physical distance, based on the recombinants we get and the phenotypes, we can easily see if they are very frequently A plus B plus C plus, the distance is really less. And they are in one phage head full. If they are rare, then physical distance is more than rare means altogether absent. It means the physical distance between A and C is, for example, more than one phage head full. We can also get A minus B minus C plus. These are rare. And this can be like, like what we saw, the double crossover kind of thing, going like this, this, and this. In this case, A plus, B minus, and C plus. This is extremely rare, they suggest. So this is like 3-point test cross in bacteria. Purpose of bringing bacterial genetics in this course is that it's a very different genetic analysis. You don't have luxury of having homolog chromosomes. You don't have luxury of, saying, okay, I set up a test cross, I set up a back cross. Bacteria simply divides by asexual reproduction. So look at the intelligence of the people who designed these tools by understanding the basic biology of phage virus, bacteriophage, how it infects bacteria, when it leaves bacteria, it in 1 in 300 phages, it may take away by mistake bacterial genome. All these were basic questions, fundamental questions, which they started maneuvering as tools to perform genetic analysis in bacteria. The next example is even more intelligent. I like these notes.
Sir, I have a question.
Yeah.
Sir, ये A, B और C जीन में ये जो थर्ड एग्जांपल है, इसमें ये भी नहीं कह सकते कि सिर्फ A जीन है आई है बैक्टीरिया में, 2 क्रॉसओवर्स होना जरूरी तो नहीं है?
No, देखो, अगर 2 crossovers नहीं होते, if we don't have 2 crossovers here and here, how A plus will get integrated into A minus? Let's say we have only 1 crossover here. How the other side, मतलब इस जगह पे crossover हुआ है, तो ये तो complete नहीं ना हुआ crossover. 2 points पे crossover होगा तो कैसेट रिप्लेस होती है, डीएनए की कैसेट थ्रू होमोलॉगस रिकॉम्बिनेशन एक्सचेंज होती है, तो एक्सचेंज नहीं ना होगी. क्लियर?
इससे जो जीनोटाइप है उसमें B माइनस C माइनस है और सिर्फ A प्लस है, तो इसका मतलब है सिर्फ A की जगह क्रॉसओवर हुआ है, बाकियों की जगह तो नहीं हुआ.
हाँ, अच्छा, ये वाला आप बात कर रहे हो. सो इसमें क्या हुआ है, एक क्रॉसओवर यहाँ पे हुआ है और दूसरा यहाँ पे हुआ है, तो A प्लस एक्सचेंज हो गया इसके साथ, तो ये बाकी B माइनस C माइनस ही रहा. डिड आई आंसर योर क्वेश्चन?
जी सर. और सर, ये हेजफुल का क्या मतलब होता है कि सिर्फ एक ही जीन ट्रांसफर हुई है?
किसका क्या मतलब होता है?
जो आप कह रहे थे हेड फुल, उसका क्या
हेड
मतलब था, वो
फुल,
नहीं समझे।
या वो मैंने आपको यहाँ पे एक्सप्लेन किया था. लेट्स गो बैक टू द फेज जीनोम. The total size of phage genome is 10 to the power 5. This is called head of phage. ये हेड है और ये टेल है फेज की. These are specialized proteins which make this capsid, जिसे हम हेड कहते हैं. Now there is size limit, and in this size only 10 to the power 5 base pairs of DNA can be packaged. We call this one head full. Clear?
जी सर.
Now when we say 1 in 300 phages by mistake can carry E. coli chromosome, how much E. coli chromosomal DNA can be packaged in one phage head? You tell me, Varis.
So 5 1 into 10 is power 5 bases long पैकेज हो सकता है.
Yes, the maximum bacterial DNA which can be packaged will be like, because there is an inherent limitation of this phage head. When this will go, and we try to see here kind of complementation, if one phage head full, let's say bacterial genome is, I try to draw here bacterial genome. This is bacterial genome. If A is here and B is here, look from here all the way here, almost 50% of the bacterial genome. Do you think this will be one phage head full?
नहीं सर.
No. So it means whenever we will say, it means A plus phenotype and B plus will never co-transduce, क्योंकि phage head full co-transduce का मतलब है एक ही phage head में पैकेज हुए वो. अगर मैं बैक्टीरिया का जीनोम दोबारा ड्रॉ करता हूं और A यहां पे है और B यहां पे है, do you think there is a possibility of one phage head full now?
यस सर, वो करीब हो गए हैं.
Yeah. So in this case A and B will always, जब भी ये वाला phage इन्फेक्ट करेगा तो A माइनस B माइनस जो है उसका फिनोटाइप A प्लस B प्लस हो जाएगा. क्लियर?
यस सर. तो सर से हमें एक रफ आइडिया मिल जाता है कि दो जीन्स कितनी दूर हैं.
एग्जैक्टली. फिर वो रिकॉम्बिनेशन भी हम देखेंगे ना कि रिकॉम्बिनेशन कहाँ कहाँ हो रहा है. ठीक है, तो ये आपको पता चल गया कि जो उसका दो जीन्स हैं वो एटलीस्ट कितना हमने कहा था, 10 to the power 5 is 100,000 बेस पेयर्स के अंदर अंदर. क्लियर?
यस सर.
डिड आई आंसर योर क्वेश्चन?
तो सर इसमें ये भी तो पॉसिबिलिटी है कि वो क्रॉस ओवर नहीं हो रहा, वहाँ पे सिर्फ एक ही हेडफुल जो था उसके अंदर सिर्फ एक ही जीन ट्रांसफर हुई है. तो ये भी तो पॉसिबिलिटी है इस एग्जांपल के अंदर.
हाँ जी, मैंने आपको एग्जांपल दी है. अगर एक ही जीन होगा, लेट्स से इस वाले सिनेरियो में एक ही अगर पैकेज हुआ है, ठीक है, तो जब वो ए माइनस बैक्टीरिया के जीनोम के साथ फेज का डीएनए आ गया जिसमें एक ही हुआ है, तो ये कितना डीएनए, यहां से लेके यहां तक डीएनए का साइज क्या है तकरीबन फेज का
10 raised to power 5.
तो रिकॉम्बिनेशन तो होगा ना, यहां हो जाएगा, यहां हो जाएगा, ये एक्सचेंज हो जाएगा, तो आपको सिर्फ A प्लस मिलेगा, B प्लस नहीं मिलेगा. डिड आई आंसर योर क्वेश्चन?
यस सर.
गुड, गुड क्वेश्चन. आई होप मेनी अदर्स आल्सो गॉट क्लैरिटी, अगर वो फंसे हुए थे तो. यहां तक क्लियर हो गया?
Yes, sir.
यार वायरस के अलावा भी कोई बोल दिया करो. वायरस की तो मजबूरी है बोलना क्योंकि वो वायरस है. तुसी भी कोई बोल दिया करो बाकी सारे हैं. मुझे क्योंकि ब्लैक बॉक्स मेरे सामने इस टाइम से ब्लैक बॉक्स है. तो आवाज आ जाए तो मुझे in the classroom I become confident because I look all around and I can see through your eyes who is with me and who is not. And here the problem is I'm talking to a box. So just like TN5 mediated mutagenesis, we said everything has limitations. You know, nothing is solid, perfect. Every rock solid tools, you know, because these are generated by humans, we find some limitations. So transduction also has its limitations, because in case of transduction, for example, here we said we are going to select for A minus B minus, A plus B. It means we have to have very good selectable markers. How we will know it is his plus? How we will know the transduction, not his plus? Let's take one step back. If the transduction is successful, if we can select for his plus, it's a good phenotype to screen. But let's say we don't have good selectable marker for phenotype selection. How we will know A is transduced or no? So one of the major challenges or limitations of transduction is lack of good selectable markers. However, they took one step more and they said, okay, you know, we can use our TN5 mutagenized mutant population to perform transduction and then transduce and select for kanamycin. Did you get it what I'm saying?
No, sir. Could you repeat that?
So what we have, we had a mutant population of bacteria in which genome was mutagenized by TN5 transposon, and all these bacteria were kanamycin resistant. ये तो याद है सबको? 2 into 10 to power 4 ये population हमारे पास थी.
Yes.
To, you know, overcome this problem of good selectable marker, said we can generate phages, use TN5 population, mutagenized population, which are all kanamycin resistant. And we know these, how much there will be, 40,000, I think.
So 20,000.
20,000.
20,000, sir.
20,000 bacteria carry independent TN5 mutagenesis. अगर मैं bacterial genome को draw करूं, इधर कर देते हैं. We have independent TN5 insertions. Independent, each line means independent kanamycin resistant bacteria everywhere in the genome. If we use this one, now this bacteria, and we do transduction. We take a bacteria. There's very little space on the screen.
So wait, I'm a little confused. Are we using a bacterial chromosome which has kanamycin resistance
Yes,
gene?
yes, yeah.
Right?
So we are going to
Okay.
use TN5 mutant population, and we infect them with phage. And when phages will leave, 1 in 15,000 phages will carry by mistake bacterial genome. Is this the probability? Are you all
Yes,
with me?
sir. Yes, sir.
Yes.
Now these phages,
Yes, sir.
we select also for kanamycin. Because TN5 is randomly integrated, and if we have are phage kanamycin resistant, it means TN5 is a small gene. In one head full, we have TN5 transposon, which will help us in selecting phage DNA, plus the other bacterial genome.
Sir,
Yeah.
but if we're saying, let's say अगर हमारी 20,000 की population है of mutagenized E. coli, so and you're saying that 1 out of 15,000 phages is going to carry the bacterial genome. So how will we isolate one phage? Won't that be practically impossible?
No, not impossible. Phages are microscopic particles. Okay, we grow bacteria, lawns of bacteria, and these, just like the bacteria, we spread on the plates, and we throw, let's say, 10 to the power 9 or whatever phage particles on this plate, and we spread it. Now, in the morning when we come, just hold on a moment.
Oh, okay, wait, sir, I got it. I think I was getting confused. I was thinking कि मतलब 20,000 bacteria से सिर्फ एक phage होता है.
Let me come.
You're talking about the number of phages, right?
Yeah.
I think I got.
So what we do, what went wrong? So what we do normally, we have spread here bacteria. Okay. And these bacteria uniformly spread सारे इस पे. Now we take soft agar. Soft agar means it's not hard, but थोड़ी concentration होती है. उसमें हम phage particles को mix करके, क्योंकि phage particles को थोड़े से microliter में millions आ जाते, हम वो phage particles को इस पे spread कर देते हैं. Soft agar में मीडिया होता है, LB मीडिया या जो भी मीडिया आप यूज कर रहे हैं, प्लस very little amount of agar, and हम इसके ऊपर उसे spread कर देते हैं. तो दो layers हो गई. एक लेयर में क्या हुआ, आपका बैक्टीरिया सारा है, और दूसरे में आपने फेज जो है जी वो स्प्रेड कर दी. नाउ व्हाट विल हैपन इन द मॉर्निंग व्हेन वी कम, फेज, अब बेसिकली फेज क्या हो गया, वो बैक्टीरिया के ऊपर उस एनवायरनमेंट में आ गए. इन द मॉर्निंग व्हेन वी कम, वी सी clear zones, very clear. And there are turbid regions. Turbid nahi sahi lafz. Translucent. Do you know translucent?
Yes, sir.
In which is, bacteria is grown, a lawn of bacteria is grown. Light उसमें से नहीं pass करती. Translucent यही होता है? Am I right?
Yes, sir. It's a semi.
Sir, no, sir, opaque.
Opaque, yeah. So these regions are called plaques. These are clear. They are not opaque. And these ones are opaque. The ones which are opaque or translucent, their phage has not infected bacteria. But these plaques are the ones where there was a bacteria, got infected by phage, phage lyse this, infected next bacteria, next, and the whole region is due to lysis of bacteria. Now this one plaque is actually due to infection of a phage. You pick this plaque. This is representative of single phage population. Okay. This one is another one. This one is another one. This one is another one. And then you isolate their DNA, or you take this plaque number 1, phages from here, infect A minus B minus, plaque number 2, infect A minus B minus bacteria, and that's how you isolate phage particles. Clear? Did I answer the question?
Yes, sir. Yes, sir.
Okay. Now, let's overcome all these limitations we said and do another screen, which I loved when I read this, because I'm not a bacterial geneticist, let me tell you. I only read this when I started preparing for this course 10 years ago. And I could not help appreciating the intelligent mind, whoever did this or whoever designed these kind of already bacterial genetics. I think this is a miracle of human mind. Extremely ingenious thing it is. Um, I never thought of metadiploids and doing dominant, recessive, complementation analysis, etc. But this screen is even more interesting. The next one. Let's say we are interested in bacterial motility. Bacterial motility. Bacteria has flagellum, and this whip-like structure goes like this and helps in motility of bacteria. And this whip-like structure is of course called flagellum, and we owe to the intelligent people of bacterial community who has actually told us that flagellar proteins are just like molecular motors. They are proteins which rotate the whole flagellum like this, just like a mechanical motor. And when they wanted to understand the motility, they said, okay, let's find mutants which are not motile. So it means the phenotype will be non-motile bacteria. This phenotype is easy to screen because these bacteria are not able to move. If you have, I just gave you example of, where's that slide, the blank slide I added. Here. You can plate bacteria. And then you overlay with soft agar, which contain nutrients. So the bacteria from bottom layer will move up. Since you will get almost, because there is a limitation of mutagenesis. It's a random process, and you already saw one in, I don't know how much was the number of mutants, even if we use TN5. One in 10 to the power 5, I think. Okay. So it means majority of bacteria will move up for nutrition, and the ones which will be non-motile phenotype, we won't be able to see them. Are you getting the question I'm trying to phrase to simplify it?
Yes, sir.
I wish I am a good at drawing, and then I can draw some 3D. So let's say here, let's say we plated bacteria here like this. It's a lawn of bacteria, but we plated like this, or even we spotted them like individual colonies, 100 on each. Then we overlaid them with another, and all of them, if they move, we won't be able to screen among millions of bacteria, we won't be able to see non-motile because all the others move and they make lawn. This screen is very interesting because they said, okay, we can use, in this case, instead of mot A minus mutation, because we cannot screen for immobility of bacteria. So they use this TN5 population, phage DNA, which by mistake took away this bacterial genome, which we calculated 1 in 15,000. And then they said, instead of screening for what we said here, we screen for, the mobility of our mutants, new mutants. What we are going to do now, we are going to use mot minus mutants, which exist already. Let's take mot minus mutant. And this mutant we infect with phage, which we grew on TN5, which are basically kanamycin resistant bacterial population. And this population was wild type bacteria. Two things. Wild type bacteria for motility gene. Otherwise, they are mutants. क्योंकि 2 into 10 to the power 4 bacteria अगर infect किए हमने phage से, which already contain TN5, तो वो तो mutant है. But they are wild type for mot plus gene. Now, these phages, one headful, if there is a TN5, one headful lying, so TN5 lying next to mot plus, so possibility is in this genome, we have kanamycin resistance plus mot plus gene packaged. Now we infect our mot minus mutants. So we grew mot minus mutants. And we infected them with all the phage particles we selected one by one. And what we did, if phage on this lawn, I made a mistake. So this was a bacteria, because all of them are mot minus. The lawn we drew is mot minus, immobile. In the soft agar on this lawn, we layered with these phages, which we generated from TN5 mutant population. Now, if on this lawn, I try to draw here now, this lawn, if one of the phage particle which contains mot plus gene has infected this bacteria, mot plus came on the phage genome, bacterial genome was mot minus. Now, this bacteria will be able to move. Soft agar and all the others which are mot minus in the lawn, they are not able to move. So what we have done, millions of bacteria which are mot minus, only the ones which will get transduced with the phage which carry TN5 next to motility gene, only that bacteria will move and come on surface and will start growing there on the surface of the plate. and we will isolate, we will know this phage, we'll know this bacteria. We can, since we know TN5 DNA, we can go back to the original plaque from where we isolated this phage, and we sequence the gene or the DNA sequence packaged in this phage particle. And we will know that which gene, TN5, which genes, because this is one head full of phage DNA, what are the genes and which one is causing MOT minus bacteria to MOT plus, because these are wild type genes. TN5 is just being used as a selectable marker lying next to these genomes. Did I explain it correctly? Is there any question? Clear?
Yes, sir.
I
Yes, sir.
explain you
So-
once more. I explain you what is written in your notes. So you will start with TN5 insertion of E. coli, which is wild type E. coli, MOT plus, it's not a mutant. You grow minimum 2 into 10 to power 4 phages. You remember why? Okay, on this is what the population was. 2 into 10 to power 4 was the TN5 population. And then, and again, I would like to emphasize this strain of bacteria which carry TN5 is wild type for motility gene. Now you use the P1 phage, which grew on this strain, all the P1s which grew here. You infect the motility minus mutant strain and select for transduction of kanamycin. That will be due to presence of TN5. You will get many TN5 positive, because let's say we got 1 in 15,000 here phages, and many you isolated, they will carry 15,000, they will carry kanamycin resistance. But once you will screen for motility of the bacteria, like I just explained you, so each of the TN5 positive, which means each of the kanamycin resistant P1 phage, which carried one head full, each of them you select on MOT A minus 1 by 1, and you screen for co-transduction. This is very important. Co-transduction of TN5 together with motility gene. And how you screen for motility, I explained you that. And co-transduction means when we will select for kanamycin resistance, we will select for motility of bacteria, just like I explained you through soft agar, both the phenotypes. Given P1 phage, one head full corresponds to this much genome. If TN5 is next to mobility, there will be co-transduction. Bacteria will be motile, as well as bacteria will show the kanamycin resistance. And you will go back to the original plaque of TN5, and you will isolate gene I explained you. That's the discovery of new gene for motility. Is there a question?
Sir, I have a question.
Jivares?
सर तो हम स्टेप 1 में बस अपना टाइम ही ज़ाया कर रहे हैं, हम डायरेक्टली इसका मौत ए के लिए क्यों नहीं स्क्रीन कर सकते? हम स्टेप 1 में से जानने की क्या ज़रूरत है?
हम डायरेक्टली इसको मॉड ए के लिए क्यों नहीं सिलेक्ट कर लेते? कहां पे डायरेक्टली सिलेक्ट करें?
ये हमने पहले TN5 की इंसर्शन ई कॉली के लिए हमने यूज की थी,
या
तो हमने P1 फेजेस के लिए हम करते ही ना, हम डायरेक्टली इसका मौत ए के लिए, क्योंकि फेजेस हमने पहले से मोट ए प्लस की जीन है तो हम डायरेक्टली उनको यूज कर लेते स्क्रीनिंग के लिए, हमने वो स्टेप 1 में से क्यों गए हैं?
Just hold on. Let me add a slide here so that we. Okay. Can you see my slides?
यस सर।
So what I understood is Varis is saying if we have which population should be selected for mort minus phenotype directly?
हमने जो TN5 इकोली को यूज किया था उसकी मुझे समझ आई, हमने उसको क्यों यूज किया था P1 फेजेस के लिए। हम डायरेक्टली इनको स्क्रीन कर सकते थे मॉट ए या मॉट माइनस जीन्स के लिए, हमें वो स्टेप 1 में से जाने की क्या जरूरत थी?
Okay, I
क्योंकि
yeah.
सो क्योंकि हमें इनिशियली तो ये जब स्क्रीनिंग हुई थी उस वक्त जीन्स नोन नहीं थी, इसलिए वाइल्ड टाइप बैक्टीरिया को यूज किया गया था टू आइसोलेट द जीन्स, और उनको फिर मोटिलिटी जो म्यूटेंट्स थे उनको ट्रांसड्यूस किया था यूजिंग द फाजेस, जिनमें हमारे पास मोटिलिटी वाले जीन्स आ गए थे। सो इनिशियली दीज जीन्स वर नॉट नोन, ठीक है, पहले नोन नहीं थे, इसलिए हमें उसमें वाइल्ड टाइप में से उसको आइसोलेट करना पड़ा यूजिंग द फाज। सर ओवर टू यू।
Thank you. So Varis, if I phrase your question and try to explain so others also understand. So I think you are asking why we cannot directly screen TN5 population for motility. This is the
यस सर।
question.
यस सर।
No, we can. I did not say we cannot. Look, you have TN5 population, then, and I explained you the limitation as well. Let's say now on the bacterial genome, we have this, the population we made TN5 population 2 to 10 to power 4, for which we had nearly 100 plates बनाई थी ना हमने colonies की, ठीक है? Now before we go to वो जो हमने की थी ना 100 प्लेट्स अलग अलग जो बनाई थी, वो तो हमने बनाई थी because we had his minus phenotype and we इस तरह स्पॉट करके तो हमने रेप्लिका प्लेटिंग की थी, यू रिमेंबर?
यस सर।
Now selection of his minus, leucine minus, etc. is easy. So selection SA, which is key point for success of our genetic screen, is easy in such a case for auxotrophic screens, his minus or whatever nutritional deficiencies we have. Now here we have to screen for motility. And let's say we are using same population 2 to 10 to power 4. इधर जब हमने रेप्लिका प्लेटिंग की, यहां से वो स्टैम्प के साथ इधर से इधर चले गए, तो हमने तो स्क्रीन ऐसे करना जिसमें वी कैन विजुअली थ्रू आवर आईज कैन सी के बैक्टीरिया मूव कर रहा है, कोई फिजिकल डिस्टेंस उसने कवर कर दी। और द अदर पॉसिबिलिटी इज यू डिजाइन हाई थ्रूपुट माइक्रोस्कोपी ऐसे, जिसमें माइक्रोस्कोप के नीचे आप इमेजेस लेते जाओ, लेकिन यू नीड टू हैव few number of bacteria, but how you will pick up those bacteria जो immobile है, motile नहीं है, ठीक है? So let's say we use this 2 into 10 to power 4 bacteria, तो हम जब इसको select करने की कोशिश करेंगे कि ये move कर रहा है कि नहीं कर रहा, तो एक ऐसे जो मैंने आपको बताया है वो ये होता है कि हम लोग बैक्टीरिया को ऐसे स्प्रेड कर देते हैं, ठीक है, और फिर इसके ऊपर हम सॉफ्ट एगर डाल देते हैं विद सम न्यूट्रिएंट, कार्बन सोर्स एटसेट्रा। क्योंकि यहां पे नीचे कार्बन या न्यूट्रिशन लिमिटेशन है, बैक्टीरिया विल मल्टीप्लाई, डिवाइड एंड दे विल मूव अपवर्ड्स। ये अगर प्लेट है, ये फर्स्ट लेयर है बैक्टीरिया की, यहां पे आपने स्प्रेड किए हैं सारे, ठीक है, ये वाली जो प्लेट, इसके ऊपर आपने सेकंड लेयर न्यूट्रिएंट मीडिया की लगा दी है, और इस वाली लेयर को मैं यूं क्रॉसेस के साथ रिप्रेजेंट करता हूं। नाउ दिस बैक्टीरिया व्हेन इट विल डिवाइड, मल्टीप्लाई, इट विल मूव अपवर्ड्स न्यूट्रिशन के लिए, ऐसे ही होगा ना?
यस सर।
When it will move upwards, so let's say हमने 2 जो भी बैक्टीरिया इस पे स्प्रेड किए हैं, तो वो तो सारे के सारे ही अपवर्ड मूव कर जाएंगे, क्योंकि मोटिलिटी जीन में म्यूटेशन होना तो फर्दर प्रोबेबिलिटी कम है ना।
यस सर।
So what we will see are all the bacteria are growing on the surface. तो वो जो मॉट माइनस म्यूटेंट है ना, वो नजर नहीं आएगा, वो छुप जाएगा कहीं, क्योंकि बाकी सारे ग्रो कर गए, उन्होंने वो उसे ट्रांसलूसेंट कर दिया, लॉन सा बन गया। क्लियर?
यस सर।
So what they did, they said we have mort minus mutant। हमने जो है ना मिल तो गए हैं, लेकिन इनका जीन नहीं पता चल रहा, क्योंकि जब हमने इसे जीन को डिस्कवर करना है, लेट्स से हमने कोई ऐसा म्यूटेजन यूज किया था EMS, या जिसने हमें मॉट माइनस बैक्टीरिया तो दे दी है, लेकिन अब हमें पता कैसे चलेगा कि ये बैक्टीरिया जो है ये किस जीन में है? क्योंकि जीनोम उसका मैंने जैसे अभी बताया 4.2 इंटू 10⁻⁶ 4 मिलियन बेस पेयर्स हैं। सो ये जो स्क्रीन ट्रांसडक्शन उन्होंने यूज की, उन्होंने कहा कि हम जो TN5 पॉपुलेशन है जो हमने रैंडम जनरेट की थी, उसको यूज करते हैं फॉर ट्रांसडक्शन। फेज जब बाय मिस्टेक लीव करेगा और 1 फेज हेडफुल TN5 तो इसमें आएगा ही आएगा, क्योंकि TN5 रैंडमली इंटीग्रेट हुआ हुआ है। तो अगर ये TN5 इज lying next to wild type gene of motility, तो जब ये फेज इन्फेक्ट करेगा मॉट माइनस को, तो इस एसे में अब क्या होगा, क्योंकि अब जहां पे हमने नीचे प्लेट किया था वो सारे का सारा मॉट माइनस है, सारे का सारा। तो जब ये जीन जो भी फेज पार्टिकल इन्फेक्ट करेगा किसी एक बैक्टीरिया को, तो वो पर्टिकुलर बैक्टीरिया मूव कर जाएगा, बाकी सारे नीचे होंगे। तो हमें पता चल गया कि ये बैक्टीरिया जो मूव अप किया है, इसको मॉट प्लस जीन मिल गया वाइल्ड टाइप थ्रू ट्रांसडक्शन, कॉम्प्लीमेंटेशन एक किस्म की हो गई है, और इससे फिर अब हम इस बैक्टीरिया से फेजेस आइसोलेट कर लेते हैं, और हम उस फेज में जो DNA होता है उसे निकाल के सीक्वेंसिंग करवा लेते हैं, हमें पता चल जाता है ये मॉट प्लस जीन। डिड आई एक्सप्लेन आंसर टू योर क्वेश्चन नाउ?
तो ठीक है सर, तो हम बस ये पता करने की कोशिश करें कि ट्रांसडक्शन ये जो मॉट प्लस जीन है ये TN5 के पास है या नहीं, तो हम डिस्टेंस पता करने की कोशिश कर रहे हैं।
नहीं, डिस्टेंस नहीं, TN5 सिर्फ यूज हो रहा है एज ए मार्कर फॉर सिलेक्शन, क्योंकि इधर जब ये मूव करेगा तो केनामाइसिन रेजिस्टेंस भी होगी, ठीक है। अब केनामाइसिन रेजिस्टेंस तो हर उस फेज में होगी ना जो फेज हेडफुल TN5 के रीजन से लेके गया, लेकिन ऑन टॉप ऑफ दैट जब हम मॉट प्लस के लिए भी स्क्रीन कर रहे हैं, मोबिलिटी के लिए भी यहां पे, तो अगर वो मूव कर गया है और साथ केनामाइसिन रेजिस्टेंस है, तो हमें पता चल जाएगा कि इस वाले फेज ने यहां से वाइल्ड टाइप फेज TN5 was lying next to mort plus gene. Clear?
यस सर।
So TN5 here is being used as a selectable marker या assay.
ठीक है सर।
Anyone else? Oh, time is up. Anyone else? कोई और question? आपका next exam is due after the next lecture, I believe. ठीक है? So in the next lecture, we'll have another quiz. आज क्या दिन है?
Sir, आज Tuesday.
Today is Tuesday. So next lecture में हम यूं करते हैं कि हम सिर्फ और सिर्फ माइक्रोबियल जेनेटिक्स का जो ये आपने किया है आज तक का, आज दूसरा लेक्चर है, आपने 2 लेक्चर्स कर लिए माइक्रोबियल जेनेटिक्स, इन 2 में से क्वेश्चन आएंगे, ठीक है, क्विज के।
Sir, can we please have it on next Tuesday? We have a lot of midterms this week, and we just
Yes, sir. This week, yes, sir. This week is totally packed.
अच्छा, मिड टर्म स्टार्ट भी हो गए हैं?
Yes.
Yes, sir.
चलो ठीक है, हम नेक्स्ट ट्यूसडे को कर लेंगे, नो प्रॉब्लम है। तो अब तक
Thank you.
माइक्रोबियल जेनेटिक्स भी कंप्लीट हो जाएगी और फिर हम तीनों लेक्चर्स का कर लेंगे ताकि आपकी एक किस्म की रिवीजन भी हो जाए।
Thank you, sir.
ठीक है, एनी अदर क्वेश्चन? Any other question? If there are no more questions, should I say goodbye then? Allah Hafiz.
Yes, sir. Allah Hafiz.
सारे चले गए जो 3 बंदे हैं यहाँ पे। अल्लाह हाफिज।
जी सर, अल्लाह हाफिज।